And our next speaker is Ralda Nehme, who is a group leader at the Broad Institute, or specifically at the Stanley Center there for Psychiatric Research. Ralda, take it away, please.
Thank you so much for this introduction. It's really exciting to be here today. Lots of exciting talks, lots of familiar faces that I'm really glad to see again. So today I will tell you about our efforts to investigate human cellular phenotypes that are associated with the 22q11.2 deletion in the context of the neuropsychiatric symptoms that are seen in the patients. So my lab is really interested in understanding how the genetic variants like 22q11.2 deletion that have been associated with psychiatric disorders, how these translate into cellular changes that affect cellular processes and eventually the brain function and might lead to disease in some individual. So really what we're interested in doing is bridging this variant to function gap. And this slide has been shown already earlier today, but if we take a step back and think about the genetic architecture of complex disease and specifically schizophrenia, we can appreciate that a lot of genetic variants have been identified that increase risk for schizophrenia. And these range from common variants that are frequently present in the populations. These are typically identified via genome-wide association studies. And then on the other end of the spectrum, there are the rare variants that are infrequently present in the population and confer a high risk for disease. So today I'm going to tell you about our efforts, focusing specifically on 22q11.2 deletion, but I really want to highlight that in the field overall, it is unclear if these common and rare variants, if they act via shared or different mechanisms to lead to psychiatric disorders. So needless to say this, there's no need to introduce the 22q deletion syndrome to this audience, but I want to highlight here that the fact that this deletion syndrome occurs at a high enough prevalence to be able to acquire a lot of samples is actually quite beneficial for functional studies, because this enables us to achieve statistical significance in our functional studies. And the other thing I want to highlight is that this deletion syndrome, as has been discussed many times already, is not only associated with schizophrenia, but with many others, neuropsychiatric and neurodevelopmental disorders. I think Raquel and others have alluded already to this fact that this disorders actually manifest in different ways. And if we take at the diagnosis that are most common during childhood, these are more typically autism and ADHD, whereas in adults, we see mood disorders and schizophrenia more commonly. So this then begs the question of how can a stereotypic deletion result in different diagnoses and different ages of onsets in different individuals. And so we then wondered if we could kind of examine in an unbiased way gene expression in individuals and during brain development in different individuals. And again, to recap where the field is right now, that as you guys know, this deletion comprises many genes and the 3 megabases that are more commonly deleted. And it has been studied over many years. However, if I may say, there's no single gene in this region that has been associated that can explain all of the neuropsychiatric phenotypes. And then this has been later on confirmed by the genetic studies, whereby no single nucleotide polymorphism, either common or rare, has been identified to be associated with the neuropsychiatric phenotypes. And again, this is not surprising because we know that most of the common variants that are identified to be associated with psychiatric disorders occur in non-coding regions, so 90% of them. So again, this is why we decided to take an unbiased genome-wide approach and ask, what is the effect of this deletion on gene expression during brain development? And to understand how the same deletion could lead to different diagnoses and different ages of onset, so it was important to take into consideration both cell type, but also cell stage. And the aggregate of genetic studies so far seem to implicate neurons as a cell type of interest in psychiatric disorders, and specifically excitatory neurons have been implicated in schizophrenia heritability. And fortunately, we had recently developed quite an efficient approach to generate excitatory neurons from stem cell. And this leveraged the neurogenin 2 overexpression approach that has been identified by the Sudoff and Werning labs, and combines it with another strong neuralizing factor, which is the inhibition of SMAD and Wnt signaling to pattern towards a forebrain fate using small molecules. So with this approach, we can generate homogeneous populations of excitatory neurons that are electrophysiologically functional in 4 weeks. And so we assembled a cohort of patients that are individuals that are either unaffected or have no symptoms that are diagnosed, or individuals that carry the 22q11.2 deletions. So our cohort size was 51 individuals in total. We generated stem cells from each of these individuals and then generated neuronal progenitors and excitatory neurons. And then we examined gene expression changes in cases versus control in each of these 3 cell stages. And this was work that I began as a postdoc in Kevin Egan's lab and continued in my own lab. And I want to highlight contributions from Olli Pietiläinen, who is a great statistical geneticist who's really contributed a lot to the analysis in this project. So when we compared gene expression changes in cases versus controls, and I'm showing you here the changes in neurons and the neuronal cell type, we saw that we could identify a robust decrease in the genes that are located within the 22q region. So these are indicated by red dots right here. But we also saw that there were genes outside of the deletion region that were significantly changed. And I want to point out here that it was important for us to have a kind of powered sample set, enough samples to be able to detect even the significant change in the deletion genes. So when we conducted a pilot study with just two cases and 2 controls, all of the red dots, or most of them, were blue. So we were not powered enough to see these changes with a small sample set. And again, this is something important to keep in mind when we're conducting functional studies. So what we found when comparing all of the gene expression stage changes across the cell states is that the genes that were located inside the deletion region, or the cis genes, these were changed in mostly all of the cell stages, which was not surprising because we were missing 1 copy of these genes in the deletion cells. However, what we found is that the gene expression changes, the genes that were in trans outside of the deletion region, were really unique to each stage, to each developmental stage. Only 1 gene outside of the deletion region were changed across all cell types. And so from there, we wondered if these genes that were changed in transcript abundance could be the same genes that have been implicated by the broader studies of autism and schizophrenia. So first, we looked at the genes that have been implicated in early neurodevelopmental disorders, such as intellectual disability and autism spectrum disorder. And what we found is that the genes that were differentially expressed, specifically in the progenitor state, these were enriched for interactors with proteins that have been associated with autism spectrum disorder. So this enrichment was specific to the progenitor stage, and it was also specific for neurodevelopmental disorder. We did not observe this enrichment when we looked at inflammatory bowel disease genes, for example, or Parkinson's disease genes. And the enrichment disappeared once the cells became neurons. Now, since the excitatory cell identity have been associated with schizophrenia, we also wanted to look at schizophrenia heritability. And so what I'm showing you here is some traits that have been, for which summary statistics exist in the UK Biobank. So genome-wide association studies have been conducted for each of these traits. And when we looked at the aggregate of these traits and the genes that are differentially expressed in our cells, we found that there's specifically an enrichment for schizophrenia heritability, and that signal was specific for neurons. So we did not find an enrichment in the earlier stages in the progenitors or stem cells. And we were able to confirm this result using 2 other methods, LG score regression analysis and magma analysis. And so these again are the genes for which common variants have been identified by the schizophrenia GWAS studies. And not only did we see an association with the genes with common variants in schizophrenia, we also found an enrichment in heritability for the genes for which rare variants have been identified by the Schema Consortium, or the rare variant exome sequencing study of schizophrenia. So as you can see here, specifically again in the neuronal cell state, but not in the progenitors, we can see an enrichment for genes for which loss of function mutations have been identified in schizophrenia patients. So from there, we wondered what neurobiological processes do these changes in gene expression might they impact? And to get to this question, we built this kind of tool to build a protein-protein interaction network that not only takes into account the interactions between proteins that have been published and validated in the literature, but it also takes into account the p-value of the differentially expressed genes. And I'm showing you here the network that we built for specifically the neuronal cell type. And there we found that there were three main pathways that were changed in the neurons: the presynaptic pathway, the proteasome or ubiquitin-dependent pathway, and the June-Fos gene activity-dependent, gene expression activity-dependent pathway. And for each of these, we were able to validate the changes by an orthogonal method. For example, SynGO analysis or synaptic gene ontologies. In the case of presynaptic gene products, Go analysis for the proteasome, and motif enrichment analysis for the activity-dependent gene expression pathway. So given that the June-Fos activity-dependent gene expression pathway is known to play a role in reducing synaptic transmission upon sustained activity, this led us to wonder whether the transcriptional activation of this pathway could result in decreased network activity in neuronal cultures with 22q11.2 deletion. So we looked at the physiology of these neurons using multi-electrode arrays, which are cell culture dishes, which are coated with tiny electrodes that enable us to measure the electrical properties of the neurons. And in that way, we were able to see that indeed there's a reduced network activity in the patient neurons that we could detect starting at kind of 21 days or 3 weeks in culture. And this electrophysiological change is indeed consistent with the changes that we could see at the transcriptional level. To confirm these results and demonstrate that it's really the 22q11.2 deletion that is responsible for these changes, we deleted the 22q11.2 region in an otherwise neutral genetic background using CRISPR-Cas9. So here we designed guide RNAs that target cut specifically and the LCRs that we just heard about. And we chose a sequence that was exactly the same in A and D, so that it would cut in both places. And we validated the presence of the deletion in the stem cells, the isogenic stem cells, by using FISH analysis and also other methods such as digital droplet PCR and SNP array analysis. And in our isogenic cell lines, what we found is that, not surprisingly, we could see that the 22q11.2 deletion genes were indeed reduced in the deletion lines. Again, not surprising, but what was very exciting is that we also could see that we could reproduce the changes in trans outside of the deletion region. So we had a high correlation of the changes between our discovery set and our isogenic set, including the kind of synaptic ontology enrichment, and 76% of the changes overall were in the same direction. And we could see that there's indeed an enrichment of genes and with presynaptic ontologies, ubiquitin pathway, and June and Fos targets. So the last thing we did here is that we also wanted to check the changes at the protein level. So with a subset of our case control samples, we also performed whole cell proteomics on the neurons. And there we could see that there is a reduction of specifically presynaptic proteins with kind of annotation in the SynGO software that has been developed by Matthias Verhage and his group, that it's actually we found to be a really useful analysis tool. And many such presynaptic proteins can be really interesting. For example, Nerexin-1, synaptotagmin 11. But here I want to highlight specifically one particular one, which is SV2A, or synaptic vesicle glycoprotein 2A, which these protein levels of SV2A are downregulated in the patient neurons, and we could validate this by immunostaining. And this caught our attention because SV2A has recently been shown to be reduced in schizophrenia patients, and there are rare variant associations in this locus that have been linked to schizophrenia by a couple of studies. And so SV2A constitute a really promising biomarker, so there's a PET ligand for it, not only for schizophrenia patients, but also for the 22q11.2 patient population as well. So to sum up kind of this part of the results, I've shown you that the 22q11.2 deletion confers cell type specific changes in gene expression. During earlier developmental changes, we could detect the regulation of genes that are implicated in autism and intellectual disability, whereas during later developmental stages, we could detect the regulation of genes that are linked to schizophrenia. I've also shown you that this deletion may be acting through the Jun-Fos transcriptional pathway to regulate protein homeostasis and presynaptic function, and that we could validate these results in isogenic cell lines. We're now exploring additional phenotypes linked to 22q11, such as chromatin organization, cell morphology, and cell function. But I kind of wanted to come back here to my earlier slide and say that we think that our results suggest that there's convergent biology between these rare and common variants that are implicated in schizophrenia. And so with this, I want to kind of take another step back, and hopefully I've convinced you that human pluripotent stem cells can be really useful to understand human disease, and that they can provide access to the human tissues that are affected in disease. And from human pluripotent stem cells, we can make not only neurons or brain cell types, but also any other cell types, like we heard earlier from many people. And for this reason, we really decided to expand on the catalog of human stem cells that are available for research and that are well characterized and QC'd. And one area that was really important to us is to acquire not only like the cellular resource, but also to generate genetic sequence, such as whole genome sequencing data from every single stem cell line that we have, so that in downstream experiment, we could link this genetic sequence to the cellular phenotypes that we observe. So through a large collaboration, we've collected samples from many different individuals and then expanded them, QC'd them, and cryopreserved them in-house, and performed whole genome sequencing and SNP array analysis on them. And then here, just very quickly, I'm showing the breakdown of diagnosis in our collection. So we have control samples, but also samples from many individuals with psychiatric diagnosis. And then importantly, I want to highlight that we are starting to mirror the representation as far as ancestry in the US Census in our collection. However, I don't think we're doing well enough yet. I think we can do better, and we should do better. And this is for many reasons. So if we think about the genetic studies, it is well accepted in the field right now that there's a strong bias when it comes to ancestry and genetic studies. For example, while European individuals make up 16% of the population worldwide, they make up 79% of GWAS participants. And unfortunately, this bias is also mirrored in existing iPSC collections. If we look at the collections in the US and Europe, we can see that 71 to 91% of individuals are made up of European ancestry individuals. So I think that's really important to diversify our stem cell collections for many different reasons. And this is because iPSCs can really capture the genetic heterogeneity across human population. And this genetic background is why it's really, we are personally attracted to human cellular models. We can't have human genetic backgrounds in other model organisms, for example. So we can generate cells from ancestrally diverse individuals, stem cells, and then differentiate them into many disease-relevant cell types. And then this will really enable the assessment of variants, the functional assessment of variants that might not be present in European populations, that might perhaps only be present in some other populations and be linked to disease. There's also very little information about drug efficacy and safety in non-European population that is well documented, that many drugs have different responses and side effects and different that are ancestry driven. And most clinical trials are currently, they take place in either the US or Europe. And as a result, there's a lot of toxicity that is reported outside of these populations. For example, in South Africa, 1 out of 12 of the hospital admissions are not because of a disease that someone is sick with, but because of side effects to the treatment they're receiving. So we think
Ralda, about 2 minutes, okay.
All right, I think 2 more slides. So, excellent. Yeah, great, thanks. So we can really leverage stem cells to be able to better understand how human genetic variations is associated with specific cellular phenotype and how these might be linked to disease, and also to develop really tailored treatments that are not only applicable to only one part of the human population. And so how might we begin to kind of invest in making resources that are diverse? And there are many different genetic efforts now that are global, that are taking place, that can be leveraged to also derive stem cells in parallel and without having to redevelop the infrastructure. One example is the NeuroDev study in South Africa and Kenya that is led by Kirsty Donald and Elise Robinson, and we're starting a collaboration with them to also be able to derive stem cells. Actually, 95% consent rate of individuals in South Africa, for example, for stem cell derivation, which is great. Another large initiative is taking place in Singapore through their National Precision Medicine Program. Again, and they want to incorporate stem cell component there, and the population in Singapore is particularly attractive because it represents 80% of the diversity in Asian populations. And so with this, I want to thank the people who did the work, people in my lab, and our wonderful collaborators, funding sources, and of course, all donors and their family. Thank you.
Fantastic. Thank you very much. Thank you. Thanks, Ralda, and thanks to all the speakers. We are not that much behind schedule, and we have a break coming up, so we can spend at least, I think, 5 minutes or so. We eat a little bit into the break time. Anybody who is interested, please stay on, and we have some questions. I'll go through questions first that have accumulated in the various chat and message boxes. There is one question. Let me see, where is it now? I have to scroll. There was one for Casey. Where is it now? Oh yeah, here from Peter Scambler. I'll just read it in the interest of time. Casey, Peter writes, the son died. Were there any Xlinked variants that might contribute to this?
Yeah, that's a great question. Unfortunately, the DNA quality was really poor, probably because it was preserved in an FFPE block for many years. And so we found way more variants in his exome than you would expect in an individual. And so we weren't comfortable moving forward. We couldn't disentangle, quality issues from true variation. And so I don't have a good answer, but I think that's a good hypothesis that it could have been Xlinked. Yeah.
Right, thanks. And then let's see, now there's a different window here. There is one for, I think this is for Joris. Well, I'll read it out and whoever wants to answer. There's a question, if the LCR reflects a transposable element, this is from Nikolai van Eers, are there fragments of these LCRs on other chromosomes?
Yeah, maybe my slides give a little bit the wrong impression. There's transposable elements within the LCRs, but there's also regions which are quite unique, and large blocks of quite unique, meaning that they are presented 2 times, 3 times, maybe 8 times on the other LCR blocks in 22. But interspersed with that, you have the HERVs, the lines, the signs, and some other transposable elements. Obviously, the transposable element families are present at all the chromosomes, but some of these more rare blocks are unique to 22. So the expansion of the LCRs in 22, we still don't understand really how these blocks have arisen. There are some hypotheses indeed that there are transposable element-driven translocations, and then by jumping, they captured some of the sequences of one locus into another. And then stabilized over during a hominoid evolution. But yeah, maybe that's providing an answer to that. But,
thanks, Joris.
Yeah.
Let me insert a question of my own, because you just mentioned it just fits here. Do marmosets have those in their genomes, or it's really only the higher order primates? I should know this, but I always wanted to look up.
I should know, yeah. I don't know, maybe, no. Maybe.
Because the genes are very synthetic in the marmoset genome. That I did look up, but I don't, okay, good. Well, I feel less bad for not knowing it. If you don't know it, I feel free to, okay. And then, Ralda, well, first also a self-serving question. How do we get our hands on some of these 800 cell lines? We just fill out a short form, almost no paperwork, and you send them to us?
Right, wishful thinking. I know, unfortunately, that's the trickiest part. There's just so many, because they are also from different countries. There's a lot of hoops to jump through, but not, I mean, there is a path to get most of them. I'm happy to send you a link, and then you can see it. So they're not obviously all 22q, right? But there are, from
how many, so about 50 of them are 22q already?
So we have 20 some that are 22q.
Okay, nice.
I
Thanks. You always had the question. Yeah, go ahead.
I have
Please.
a question to you, Ralda. So I learned from some people trying to make IPSs that it was quite hard in 22q11 patients, that they're not as easy as others. Did you experience that? Or
we have not had any issues. We had only one cell line that we had to attempt 2 times, and it was a 22q, but it also happened with other cell lines. Yeah, but in general, it went smoothly. The making the isogenic cell lines, on the other hand, that was a very inefficient process. We had to pick a lot of clones.
I was going to ask that. Yeah. Did you get a lot of off-target cutting on other chromosomes? Was that even something you could see, or do you know what happened?
We have not. I don't think, because these repeats, I don't think they are like present elsewhere when we checked, but we have not. We just did SNP array, and then again to kind of look for genome integrity that way, but we have not done whole genome sequencing post editing. Okay, nice. That was neat. Maybe 2 more questions. One more for Joris, and then I have one more for Casey. Ina Amarillo asked, how similar is this region or the 22q LCRs between Neanderthals and humans? Can we tell? I don't know if we can. What do you think, Joris?
It's a great question. Actually, we are working together with someone to look into that. But, yeah, I don't have the answer. To be honest, the short reads do not allow mapping of the LCRs properly. Yeah, it was good. Okay, great. And then Casey, I was wondering when you were talking about I think it's the NK, I forgot which, NKX5?
2.5.
Was one NKX, right? That where you have a strong heart phenotype in the mouse model at first, but then you cross it into a different background and the phenotype goes away.
Mm-hmm. Yeah.
Could that actually be, and that also reminded me of something that I seem to remember for the TBX1 mouse. Isn't there also a very strong phenotype?
Yeah, that's specific.
Has that ever been tried in TBX1 where you cross that, crossing that into different background and seeing what happens to the phenotype? Do you know? And
to my knowledge, it hasn't been done as extensively. I think, Bernice might know that.
I know the answer to that is that Antonio Boldini did that work.
Okay.
And that in C57 block 6, heterozygous TBX1 mice are more severely affected than other genetic backgrounds.
That's really interesting. Well, I wonder, could one use that actually as a discovery tool, right? Where you then sequence the genomes of these different, or you have the sequence. I mean, these are inbred strains, right? And then potentially any difference elsewhere in the genome between the 2 strains is a potential modifier. Or do you think you'd be overwhelmed? There'd be tens of thousands of variants and there's no way of sifting through them.
No, I think it's a great question. And that's something that my lab is interested in pursuing, at least in the context of NKX25. I think it's sort of similar to what Ralda was just talking about with this collection of different IPS cells that represent different ancestries, right? I think, and we're interested in taking both approaches, right? I think you can apply both to the human population as well as to different mouse strains and sort of tackle it from the same position. And looking for how genetic background is contributing based on, naturally occurring changes in genetic background. I think it's a great way to look for modifiers, actually.
Yeah. But that's really, and that's okay, great. I think, Donna, did you turn on your video because you have a question, or to tell me to move on because
no, someone asked in the chat if the cell lines are on patients, all of whom have psychiatric illness.
Oh, sorry, I wasn't watching that. Sorry, yes, right. So the question again, Greg Crabtree asked, thank you, Donna. Do all the patients, the 22q cell lines have psychiatric diagnoses?
Yeah, it's for me, I'm assuming. So the answer is yes. And most of them have a schizophrenia diagnosis with other comorbidities, such as intellectual disability. We have a couple of cell lines with just ASD or anxiety and intellectual disability, for example. But I would say maybe like 60% have a schizophrenia diagnosis.
By the way, Joris, we should add all of these to CRISPR-Catch, right? We should add these to the CRISPR-Catch cohort.
Yeah, I was thinking that, but obviously you're closest to Ralda.
But that means I have to do the paperwork. That's not a good idea, but okay. Okay.
And I'll just add that we have lots of cell lines from the 22q11 Center at CHOP, many of whom don't have psychiatric illness. And Bev Emanuel was supposed to join today, but she had a conflict at the last minute. But if people are interested in that, just give a holler.
Good,
Already
excellent.
collaborating with Yoris.
We will. Thank you. Fantastic. I thank you, everyone. Thanks again to all the speakers and to the audience, the great questions and comments. I think, let me see, what do my instructions say? So we are in the break time already, but I think that was well worth it. We resume at Raquel at
11:40. We'll
11:40. Oh, okay, good.
For session 3.
Okay, awesome. Great. Thanks very much, everybody, and see everybody at 11:40.
Thank you.